Wonderful morning or evening to all of you. I am Pradeep. I am presenting our work titled Characterization of Meditation EEG based on consistency of covariance matrices over time. My co-authors are Professor A.G. Ramakrishnan and Dr. Kanishka Sharma. This work was carried out at my laboratory, Department of Electrical Engineering, IISc, India. Meditation is an attentional regulatory and emotional training task leading to a transpersonal states like samadhi, transcendental consciousness. Based on our literature survey, there are no objective indices to quantify the depth of meditation during the practice. Existing literature on depth of meditation is based on questions on years of meditation practice, experience during the meditation recording, etc. Not many studies have been conducted in this direction to quantify the depth using an objective index. Studies have analyzed Raj Yoga practice and reported activation of many networks in the brain during meditation, increase in band power and asymmetry in long-term meditators. There are also studies on physiological changes during Raj Yoga meditation. A neuroimaging study conducted by Sharma and group reported increase in white matter integrity in corpus callosum segments in long-term Raj Yoga meditators as compared to controls. Another analysis done by us on the dataset used in the present study showed increased entropy in frontal lobe EEG. In this work, we analyze the EEG recorded during Raj Yoga meditation, which is practiced with eyes open, which makes it unique among other meditation techniques. An attempt is made to analyze the covariance matrix, which captures the interdependencies of different regions of the brain during the meditative and non-meditative states. Data was collected at Brahma Kumaris headquarters at Mount Abu. Experimental protocol consists of 2 meditation segments and 3 baseline segments recorded in the order shown in figure 1. Subjects were asked to practice 2 types of meditation for 10 minutes each. Baseline EEG is recorded for 3 minutes. At the beginning, in between the 2 meditative states, and at the end. Dataset comprised 15 healthy right-handed subjects with Raj Yoga meditation practice of more than 10 years. All the baseline segments were recorded with eyes open. In peaceful soul consciousness meditation, subjects focus their attention on the positive thought considering themselves as peaceful souls. During the angelic meditation, subjects assume to attain a blissful state. The fundamental difference between the 2 meditation techniques is the self-observing experience in peace meditation and the state of being detached from the physical body, its relationships, and the material assets in angelic meditation. The data was recorded with ANT neurosystem with 64 channel waveguard cap according to 10-10 system. Sampling rate of 1000 samples per second was employed during the recording. Post-recording, line noise of 50 Hz was filtered using notch filter followed by 0.5 Hz to 45 Hz bandpass filtering, and independent component analysis was applied on the data using EEG lab. Grouping the channels was carried out as given in figure 2 into 5 broad regions, namely frontal, frontocentral, peritocentral, occipital, and combining temporal right and left channels as temporal region. Abstract of the proposed methodology is given as a flowchart. 50% overlapping epochs of 1 second each consisting of all the channels of a lobe are considered to obtain the covariance matrices. Details of the distance between covariance matrices shall be discussed in detail in next few slides. Covariance matrix quantifies the pairwise correlation between the channels. The off-diagonal elements capture the inter-channel correlation and the diagonal elements are the variance of the channel. The proposed work is based on the distance between these covariance matrices of successive epochs, which is used to distinguish between the different meditative and non-meditative states. As mentioned in the previous slide, XIK is the kth epoch data matrix of ith lobe, where I goes from 1 to 5 as we have considered 5 brain regions or lobes for analysis. J goes from 1 to 5, representing baseline, peace meditation, intermediate baseline, angelic meditation, and final baseline segments. Covariance matrix of kth epoch of ith lobe is obtained as given in equation 1, where mu I k is the mean column vector of the kth epoch. The difference matrix is calculated between successive covariance matrices and is denoted by dik-1, where k goes from 2 to kj, where j goes from 1 to 5. Frobenius norm of the difference matrix is calculated as given in equation 3, which results in a vector corresponding to each state for a given lobe. Mean and variance of the vector obtained in equation 3 is calculated as given in equations 4 and 5 respectively. Subject 11 results using box plot without outliers of the Frobenius norms are shown in figure 4. Red lines show the median and the diamonds the mean values of the Frobenius norms of a given segment and lobe. It can be observed that the mean or median is reduced during the meditative states across all lobes of the brain. However, the trend was significant across all subjects only in the frontal lobe, as will be explained in the next slide. Wilcoxon test was used to check the statistical significance of the results obtained. Test revealed that the mean of the Frobenius norm results were significant in the frontal region of the brain. Figure 5 is the univariate scatter plot of the means for all the meditative and non-meditative states across all the 14 of subjects of all the 5 lobes of the brain, each point representing a subject in each line. Black horizontal line is the mean obtained across 14 subjects and the grey box the standard error mean. Table 1 and 2 is the z values and the p values obtained using the Wilcoxon test of the frontal lobe results. Figure 6 is the univariate scatter plot of the variances of all the meditative and non-meditative states across all the 5 lobes. Again, the results were significant in frontal lobe. A study conducted by Travis and his group reported increase in frontal EEG coherence and was positively correlated with inner orientation, emotional stability, and moral stability. The higher statistical significance in frontal lobe compared to other lobes needs to be investigated further. To conclude, the mean and the variance of the distances of covariance matrices decreased during the meditative states as compared to non-meditative states in frontal lobe. This was our hypothesis to test across all lobes, and it's revealed that the interrelationship between frontal lobe channels are consistent during the meditation states compared to baseline. This can be used to quantify the depth of meditation with an index aided by questionnaires, which is being used over the years. Our future work include the analysis of the distances between the covariance matrices in a given frequency band of interest, namely delta, theta, alpha, beta, and lower gamma bands. We thank the support extended by Spark Wing of Brahma Kumaris organization by lending their EEG system for the study and the subjects who participated in this study. I also thank my co-authors for their contribution towards this analysis and study. The following slides list our references. Thank you for listening. I shall take the questions if any.